The 2026 International Congress of Basic Science Engineering Science Night is about to begin. Please take your seats and set your mobile phones to silent mode. Thank you for your cooperation. The 2026 International Congress of Basic Science Engineering Science Night is about to begin. Please take your seats and set your mobile phones to silent mode. Thank you. Booster Robotics is a leading humanoid robotics company, with its technologies reaching universities and research institutions across more than 20 countries. This year, its robots helped Tsinghua University win the RoboCup World Championship and secured all humanoid robot football gold medals at the World Humanoid Robot Games, making it a true champion both on the field and in the market. Now, please enjoy the next show. Through these dances and different styles, we have seen how fundamental science can be transformed by engineering innovation into remarkable technological achievements. Science builds the foundation of civilization, and engineering brings discoveries. Please join me in thanking Booster K1 robot dancers from Robotics for these amazing performances. Dear guests, ladies and gentlemen, good afternoon. Welcome to the 2026 Engineering Science Night. I am host Zhang Qian from Shenzhen Loop Area Institute. First, please allow me to introduce our distinguished guest present today. Fields Medal winner, President of the International Congress of Basic Science, Chair Professor at Tsinghua University, President of the Beijing Institute of Mathematical Sciences and Applications, Professor Xintong Yao. Foreign member of the Chinese Academy of Engineering, Fellow of the Royal Society of Canada, Executive Dean of Shenzhen Loop Area Institute, Vice President of the Chinese University of Hong Kong Shenzhen, Professor Zhiquan Luo. Turing Award winner, member of the U.S. National Academy of Sciences, Distinguished University Professor at Princeton University, Professor Robert Tarjan. Member of the U.S. National Academy of Sciences, Professor at Harvard University, Professor Xiaowei Zhuang. Fellow of European Academy of Sciences, Professor at KOST, Director of the Joint Laboratory of Scientific Computing and Machine Learning at Shenzhen Loop Area Institute, Professor Jingchao Xu. Foreign member of the Chinese Academy of Sciences, foreign member of both Academies of Engineering and of Science in USA, UK, and Canada, American Academy of Arts and Sciences, Professor at Northwestern University, Professor Yonggang Huang. Distinguished Professor of Biochemistry, UCLA, Professor Julie Fagan. Senior Vice President of Jingdong, Head of AI Innovation, Dr. Xiaodong He. Fellow of the Society for Industrial and Applied Mathematics, Presidential Chair Professor of the Chinese University of Hong Kong, Shenzhen, Professor Xiaoping Wang. Welcome all. Now it's my great honor to declare the 2026 Engineering Science Night commenced. First of all, may I invite Professor Xintong Yao, the founding chairman of the ICBS, Fields Medalist, to the stage and give us a welcome speech. Thank you.
Ladies and gentlemen, welcome to China for the ICBS. This has been an exciting event. So far, we have 3 nights together. We have gathered the most distinguished minds from all over the world. It is exciting to see people in different subjects, namely in physics, in math, and engineering, come together to exchange ideas and telling each other what they think in the most modern language, including AI. And I think this is really exciting. Engineering has been one of the most important subjects in the last 10 years interacting with theoretical science, much more than ever because of the invention of AI. AI has been extremely useful and important for many areas. Even theoretical mathematics, that many important problems was either solved or helped by the idea of AI. I think this is fascinating, and this will make mathematics to change in a better manner, not in a worse manner. Many of my colleagues thought that this is a disaster, that our job will be taken over by AI. I don't believe that. I think it just helps us to get more jobs instead, because AI cannot replace human mind. The machine has a limit of what they can do. I actually had some philosophical discussion with my friends in physics, and even my friend like David Gross or Witten was worrying about what AI can do. But I explained, I said, the mystery of nature is infinite, and AI could only master a limited amount of knowledge, namely those knowledge that we know in the last 2,000 years. So I think it will never conquer human mind. We are creative, we can think, we have most emotion. All these are something that is tough for AI to take over. So I'm very happy to say that we will help by AI. We will make the science and engineers to be much better in this next 10 years than ever seen before. And I take it in an optimistic way, not pessimistic. So I hope many of our young students will learn, but learn how to think, not just using AI to help you to solve problems. We're thinking, with AI helping on the side, I think the subject will carry on in a much better manner. And of course, not just in mathematics, AI solve problems in physics, in astronomy, in many other things. And it helps. But I think next few years will be the most clear-cut problem solver by AI. After that, most problems that can be solved probably are solved. And then human being will play an important role again. But AI will certainly always be there. Engineer will be always important for us. Any technology innovation will bring science to a new platform. And we saw that in the last 2,000 years. And this, of course, happens to mathematics too, not just in science in general. In mathematics, we saw the major development in each stages. Each stage bringing really spectacular achievement. I was talking with my friends during lunch. A very simple idea, like invention of coordinate by Descartes, actually has moved mathematics in a completely new platform. Before the invention of coordinate, we only know how to draw straight lines, circles, convex, and all that. Geometry could never be moved to a new stage without understanding of coordinate. Without coordinate, Newton would not be able to apply his 3 laws of mechanics to understand the universe. So all these are concepts that was invented and make a major breakthrough and make mathematics go into a completely new level. And I think mathematics in the last century, we have seen many important major breakthrough, conceptual breakthrough. Mathematics is going on as strong as ever. So is physics, so is engineering. So I think these 3 subjects, all of our researchers in these 3 subjects come together, interact with each other, will make basic science to a new platform, will bring human beings a new understanding of nature, and engineer will bring human beings have a much better life. So I think we should be very optimistic for the future. Thank you.
Thank Thank you very much, Professor Yao. Next, let's welcome Professor Zhiquan Luo to the stage and give us a speech. Professor Luo is the Executive Dean of Shenzhen Loop Area, Vice President, and Professor at the Chinese University of Hong Kong, Shenzhen. Welcome, Professor Luo.
Distinguished fellow scientists, distinguished guests, and our friends, good evening. It's my distinct honor to address you tonight on behalf of the Shenzhen Loop Area Institute, which we also call SLAE, and the Chinese University of Hong Kong in Shenzhen at the ICBC 2026 Engineering Science Night. We are truly privileged to co-host this important event alongside with BINSA. So very happy and honored. As all of us have witnessed, the rapid advancement of AI has taken our world by storm, affecting everything we see, we do, including basic science. Yet, as we marvel at the transformative power of AI, we must also ask ourselves, what is the true engine behind, true theory behind the innovation, the revolution brought by AI? Without a deep understanding of how AI works, how the system ought to be built, we are only shooting in the dark. We are only experimenting things. We need to understand things. That's our human nature. That's what basic science needs to do and must do. And this is precisely the theme of tonight's discussion and forum and keynote addresses. Using this occasion, I would like to say a few words about the SLAE and also the Chinese University of Hong Kong in Shenzhen. SLAE is a national level AI institute aimed at training top-level, PhD-level talents in AI. So last year, we've already recruited, we started last year. We recruited more than 230 PhD students. And we also recruited more than 120 faculty members. Some of them are fractionally appointed, jointly appointed with other universities, participating universities, from top universities in mainland China and also in Hong Kong. So SLAE is actually situated right between Hong Kong and Shenzhen. It's a special area called Loop Area, borders Hong Kong. So it's the Shenzhen River bends a little bit, becomes a loop area. So that's the area we are situated in. So our goal is to integrate basic science theory and also practical experiments using large computing clusters to try and to understand how AI system works and how it ought to be built. We are developing actively theoretical foundations understanding of AI system and also the AI architecture, frameworks, algorithms, chips, and so on. In addition, we are developing AI for science and AI for engineering. We also do AI for social science, understanding the ethics, security, and so on, safety, and so on. We also have a large team working on humanoid AI, embodied AI, computer vision systems. So we have a total of more than 120 faculty members and then 240 or so PhD students. And this year we're recruiting another 300. And we're growing as we speak. We are recruiting students from all over the world, faculty members from all over the world. We are English speaking, we're international, we work very closely Most of the faculty members actually come from Hong Kong area, from Shenzhen and Hong Kong area, and students likewise. So we're recruiting all over the world. So we intend to be international, and we intend to be open. We open source everything we do. So that's where we stand. So our determination, our mission is to advance the understanding. And then our mission is to understanding of AI and developing new AI technologies. And then the ultimate goal is to train next generation AI new talents that will serve the needs of humankind, humanity. I also want to say a few words about another institution that I'm heavily involved since its very beginning 12 years ago. In Shenzhen, we established a new university called Chinese University of Hong Kong in Shenzhen. We must have heard about CUHK. That's based in Hong Kong. And yet we have another campus in Shenzhen, which is very close, 40-minute ride away from Hong Kong, just across the border. So this 12-year-old university is also international research university. It aspires to be international research university. English speaking, its academic system is the same as that of CUHK in Hong Kong. We recruit faculty members and students from all over the world. We use popular graduate textbooks or undergraduate textbooks. We produce our own textbooks, and so on and so forth. We charge tuition 20 times higher than local universities in mainland China. And yet, top students still come to CUHK Shenzhen. Top 1 or 2%, mostly 1% of high school graduates come to CUHK Shenzhen to pursue their higher education. We use the same academic degrees as that of CUHK. We are otherwise administratively, financially independent from CUHK in Hong Kong. But academically, we're integrated, same academic standards, same degrees. So we are one brand, two campuses. It's much closely integrated than UC Berkeley or UCLA, these sort of systems. We're much tighterly integrated. So 12 years in the making, the university currently is already, guess what? 14,000 plus students strong, 800 faculty members recruited worldwide. That's China speed. There's something else, Shenzhen speed. So my workload is actually 3 times at least more than that of US. I spent 30 years in North America as a graduate student first and then faculty member. So the pace of work is incredible, breathtaking. But I'm very happily working hard because I see my work having some impact on the higher education, turning out students that go all over the world to be graduate students, to be faculty members. I've recently, my first PhD student since coming back to China is now serving on the faculty of computer science at the University of Wisconsin-Madison. So I'm very happy for that. So it's a global world, and then academic basic science, we speak the same language, and then we are advancing our knowledge and the understanding of AI and other things, basic science, for the goodness of the humanity. So tonight's program on basic science engineering night promises to be rich and inspiring. Just looking at the names of the speakers, keynote speakers, we are amazed. So we have Professor Zhang from Harvard University. Also, we have a panelist, a top panelist, like from Bob Tarjan. It's like my hero when I was graduate student reading his books on data structure and so on. So amazing for the first time. Actually, I heard your talk at MIT before as a graduate student, but never shake your hand. But I'm happy to meet you here in person. but in Beijing in this event. So we also have a roundtable discussion on the future of AI and basic science. To conclude the engineering night, we also have a lovely music concert performed by faculty members and students from CUHK Shenzhen's School of Music. It promises to be very enlightening. So my sincere thanks go to Professor Yao for his vision and leadership that has made tonight's event program possible. He actually handpicked each individual speaker, each panelist. So he had proved everything, okay, of tonight's, including the performance. And also like to thank the organizing committee and all our distinguished guests and the friends present here tonight. May our discussions tonight be as enlightening and inspiring as the music that awaits us tonight at the end of the discussion. Thank you all.
Thank you very much, Professor Law. Thank you. So now we will move to the keynote report, and let's invite Professor Chen-Ye Wu on the stage to host this session. Thank you.
Thanks, Cindy. Distinguished guests, I'm Chen-Ye Wu, the Assistant Dean from the Shenzhen Loop Area Institute. I'm also a faculty member at the Chinese University of Hong Kong in Shenzhen. I'm today's host for the academic session. And every breakthrough in basic science can lead to new advances in engineering. And every advance in engineering can open new windows for basic research. So tonight, we will hear 3 talks from outstanding scientists. The topics range from biological imaging at the nanoscale to digital humans and humanoids, and then to nucleic acids and telomerase. Together, these talks will take us across different scales and different fields. So first of all, please welcome Professor Xiaowei Zhuang, member of the U.S. National Academy of Sciences, professor at Harvard University, and an ICBS Medal laureate in engineering. Professor Zhuang is a leading scientist in biophysics and imaging. Her work in super-resolution imaging, single-molecule imaging, and spatial genomics has changed how we see life at the smallest scales. Technologies such as STORM and Morpheus have opened new windows into cells, tissues, and biological systems. Her talk is entitled Illuminating Biology at the Nanoscale with Imaging. So now, without further ado, please join me in welcoming Professor Zhuang.
All right, so this works, right? All right, so I get it's a pleasure to be here again. And can I have the slides, please? All right, it's a pleasure to be here. As I mentioned that, in the morning for the medal lecture, I talked primarily about our work on genome scale imaging. And tonight I'm going to change the topic to super resolution imaging. And I guess these are the 2 areas that I got the Stephen Chu medal for. So for super resolution imaging, let's just give a little bit of a background too. As we know that our body are made of many, many cells. And inside the cells, many different kinds of molecules work in a concerted way, coordinated way to give the cell its life. And one thing that we love to do in my lab is to actually use the direct visualization approach to study how things work in living systems. And then, as I said in my award acceptance speech, that one quote that I love, which I learned from Steve, and then Steve learned from this wise baseball player, Yogi Berra, is you can see a lot by just watching. So we'd like to invent imaging approaches that allow us to see a lot by just watching. So in order to see these molecular machines work inside the living systems, inside living cells, these are some of the key requirements. First, because there are so many different kinds of molecules, we would like to have molecular specificity. Means we would like to know what are the molecules we're looking at. And then the other is we would like to have molecular scale resolution so that we can resolve them and see them, tell them apart, and then see their individual actions, their interactions. And finally, we would like to have the genome scale throughput that is so that we can see everything and then get a systems level picture. And in terms of molecular specificity, one imaging modality does very, very well, and that is fluorescence microscopy. And the reason for that is because of a lot of very important work that actually allow us to label these biomolecules with exquisite specificity. For example, you probably have heard of the green fluorescent protein, which can actually be genetically fused two proteins of interest. One a specific protein, you tag a GFP on it, then you can see it emit light, and then you can see its movement and action in living cells. And then you can also label these proteins with specific antibodies, and these antibodies could have dye labeled to it. Or if you're interested in nucleic acid, as Professor Julie Fagan, you could actually use a fluorescent dyes labeled oligonucleotide probes that all give you very, very high molecular specificity. And then in terms of genomic scale throughput, I already talked about in the morning that because of our development of MERFISH imaging, I can't believe I misspelled my MERFISH, F-I-S-H. This doesn't make sense, right? IFHS. So MERFISH imaging, we can image tens of thousands of genes, and then we can use that to identify different types of cells inside the body, different states of cells. And then here, for example, is a whole brain where we can see different types of cells now colored in different colors. So this will actually give us a systems level of understanding of the different types of cells that are existing in our body, and then see their gene expression profile and understand the molecular basis of their function. And molecular scale resolution is at least used not to be an advantage of light microscopy. And a different imaging modality gives you much higher resolution, and that is electron microscopy. And now with the revolution of cryo-EM, then you can even get atomic structure of molecules. And with the rapid advancement of cryo-ET, you begin to see a variety of different molecules in living systems and so on. But still, many molecules are still very hard to distinguish by cryo-ET. Maybe eventually we can all do that. But light microscopy remains the major workhorse in terms of imaging the molecules inside the cells, especially in living cells, because cryo-ET requires cryo temperature, so things are by definition frozen and not alive. So why is it that light microscopy, having so many good advantages, doesn't have molecular scale resolution? A key thing that we all know is because of the diffraction limited resolution, because light is a wave. As a wave, they diffract. Because of that, when you try to focus light to a tiny spot that tiny spot has a finite size. That's about half of the wavelength of light, which is about 200 nanometers or 300 nanometers, while these biomolecules are often just a few nanometers. So it's 2 orders of magnitude short of the resolution that is required to resolve the action of molecules inside living cells. So there are different approaches to break this diffraction limit and overcome this diffraction limit so that we can get to very high resolution. And one thing I do apologize is some of these things are a little bit outdated. There are some even more advanced technologies that get to even high resolution. I'll try to remember to say, but I didn't include that in the slides. The first method that actually broke the diffraction limit and then allow us to get to subdiffraction limit imaging is this approach developed by my friend Stefan Hell at Max Planck Institute. And this approach is called stimulated emission depletion microscopy, or STED, and a resolved, which is a generalized form of STED. And what it does is it uses stimulated emission depletion to actually generate a donut beam, and then hence the what we call point spread function that gets actually shrunk to an effectively much smaller point spread function. And if you use that to scan your sample and get an image, you can get much higher resolution. In case I forget, let me say that Stefan actually invented now an even newer approach called mean flux, sorry, and that actually gets to even angstrom resolution and so on. Truly remarkable approach. Now, the approach that we developed depends on single molecule imaging. So single molecule also has an image that looks like that is diffraction limited, means the image width is about 200 nanometer wide, but that doesn't prevent us from finding the center position of this image with a much higher precision. And then usually, if you're mathematically oriented, that just means that if you measure a thing, find its center, you have an uncertainty that is this much. But if you measure it 10,000 times, your uncertainty gets actually scaled down by square root of 10,000. And if it is 200 divided by square root of 10,000, you get 2 nanometer precision. And if you're not mathematically oriented, think about a mountain. And then if you have a mountain that has a certain width, you can still find the peak with a much higher precision. Now, that alone doesn't break the diffraction limit. The diffraction limit is because these individual molecules images, when they're close, they're totally overlapping. So you are not able to find the center position. So this single molecule localization doesn't work when I have a very high density. And that's often the case of a biological system. So the idea that we came up with is to say, hey, what if I don't let all the molecules emit light simultaneously? Only such a small fraction, so that their images are actually well separated. Then I can pinpoint their center positions, and then I get a next set of molecules light up. Then I can pinpoint their center positions. Over time, these center positions gets accumulated in this fashion. Then eventually, because every molecule is localized to nanometer precision, when I put them together, I get a nanometer resolution image. So this is what we call stochastic optical reconstruction microscopy. As you probably have heard of this name STORM a few times already, because people, they tell you that this is one of the things that I got the prize for. So you can see even by eye that the resolution immediately compared to diffraction limited conventional resolution is much higher. And I should say that as we publish the work, there is parallel work and of similar methods. That's by Eric Bethik and Samuel Hess. And then you can see these papers were all published actually in the same year. And actually our paper and Eric Beck's paper, they're just one day apart. So, people sometimes think alike, which is not surprising. And after that, the next thing we thought about is our world is three-dimensional. We don't want to generate a two-dimensional super resolution image. So how do we get 3D information? To get two-dimensional super resolution imaging, what we did is we determined the XY position of individual molecules. How do we get the Z information? And that actually, if you did imaging, you know that there's this focus knob. And then if you get things in and out of focus, the shape of the molecule actually change. That shape actually encodes the Z information. The downside of it is at the focal plane is the local minimum. And not only when you move above and below focus, things change, because if it's a local minimum, the slope is, see, I tend to say this because I've seen there are lots of famous mathematicians sitting here. So usually I don't go that slow on that part. So you can get this local minimum so that there's a zero slope, so you have zero sensitivity, let alone you have a degeneracy, because in above and below focus, the shape look identical. So the trick that we use, as demonstrated here, so the trick we use is actually putting a cylindrical lens. And if you're not familiar with optics, if you have astigmatism in your vision, the correcting glass is actually a cylindrical lens. And what happened is with that, the image actually turned into elliptical. And not only it made this slope non-zero to give you sensitivity, but below and above focus, you will have the image elongated in orthogonal or perpendicular directions that can give you unambiguous Z information. So this is now a three-dimensional image of this microtubule inside the cell. What I also showed you earlier when I showed you the much higher resolution at 2D, that was also microtubules inside the cell, and then you can even get the 3D information. And here is another structure inside the cell, and these are mitochondria. And I often show this image because here you can have the contrast of conventional image, 3D storm, and a section of 3D storm. You can see the major, big contrast of image resolution difference. Another reason, because this image probably showed up in many news articles and so on, because some people tell me that it also looked like some kind of a galaxy image. So. And at the time when we got the resolution, first 3D super resolution imaging published in 2008, that was 10-fold faster than diffraction limit in all 3Ds, 200 nanometer in XY and 50 nanometer in Z. And over the time, we improved that to a few nanometers, which is another order of magnitude improvement. But as I said, that the recent main flux revolution by Stefan Hell actually brought the resolution to 1 nanometer or even sub-nanometer with very high time resolution. And then storm is also a thing that we can do. Oops, this movie doesn't play. Let me see. I should be able to make it play. I guess not. Before plugging, it actually plays. It's okay. These are mitochondria that you can see them moving, fission and fusion and so on. So meaning that we do have the live cells real time resolution. All right, so over the years, we use storm to resolve a variety of, or study a variety of different structures inside the cells, ranging from simple cells such as bacteria to very complex cells such as neuron, look into subcellular structures like the synapses that are important for communications between neurons, or even our chromatin or DNA and how it is folded in the nucleus. So I'm going to then just focus on one biological application to show you that storm not only give you these sharp, beautiful images, but really lead to discoveries of structures that people didn't even know existed before inside the cell. And then that came from actin imaging. So this is a storm image of actin in a fibroblast cell, and then seeing beautiful, and this is another journalist popular images that they love, but I should tell you that they only love it for its beauty because we actually didn't learn anything new in this image. But showing that we can do that, the next thing that we did is actually imaging actin inside the neurons, because when the postdoc Ke Xu actually finished his development of a higher resolution storm microscope and then got this image, he asked me, hey, what do I look for? And I said, look for synapses in neurons, because that's where actin is supposed to be playing a very important role in memories and learning and so on, but the structure is not known. So then he did that, and remarkably, the attention is quickly shifted away from synapses, because in axons, he saw this beautiful periodic band structure. So it tells you that a great student, you tell him to pay attention to one area, he sees another thing, and then he chases this other thing. So this is a beautiful structure that is made of rings that are periodically spaced. And when I first actually presented this image at the conferences and so on, some people don't believe it. They say, such a beautiful structure in a cell as important as the nervous cell, in a structure as important as the axon, how could it have been ever missed after many, many years of high resolution imaging? Then I just show this, then they get convinced, because this is a diffraction limited image. And why? It's because the space between these rings are actually just smaller than the diffraction limited resolution. And then it's about 180 nanometers. And then also that ring to be periodically spaced also give you some clue of what other things are there, because there must be some spacers that needs to be there. And the spacer needs to satisfy two conditions. One is the length should be about 180 nanometer, and the other is the spacer should actually be interacting with actin. And that reminds us of this other structure that is known in a completely different type of cell called red blood cell. Underneath the red blood cell membrane is this hexagonal or triagonal kind of a structure, and where these arms are spectrin tetramers, and these little cylinders at the nodes are acting. And then the spectrin tetramer do interact with actin, and then they tend to have about, when stretched out, 200 nanometer resolution. So we thought, okay, that satisfies the requirement. So then we tested, we made the hypothesis that is what's connecting the actin ring. So then we tested with super resolution 2-color imaging. And then that what we do is we use antibody to recognize the middle point of the spectrum, and then also image the actin. In that case, the prediction is we should see alternating rings. And indeed, this is the beautiful alternating rings of a spectrin tetramer center and the actin ring. And then also following the leads of the red blood cell structure, we think these are short-acting filaments that should be capped by molecules tripomodulin and adenosine. Indeed, both tripomodulin and adenosine form these ring structures that coincide with actin and is alternating with the spectrum middle point ring. And then we found that this middle point is actually a platform where through the adapter anchoring can anchor many important functional molecules such as the ion channels, receptors, and so on, and to take these periodic structures and so on. So then we did a proteomic study to actually find all possible molecules that are on this. I won't go through the proteomic approach, but for those of you who know, that's basically immunoprecipitation and mass spectroscopy to get the candidates, and then using imaging to validate. And then we found all the molecules that are on this structure and how they are organized, and then these functional molecules that actually reside on the structure, using this as a platform that include, as I said, ion channels, receptors, cell adhesion molecules, and other signaling molecules. And then indeed, we found that the structure is a key signaling platform. For example, it is very important for a signaling called RTK signaling, in particular GPCR transactivated RTK. I do apologize to people who don't have the biology background, but to say that this is a very important signaling pathway. inside the cell that's important for cell survival and a bunch of other functions. And then this structure that we discovered serves as a platform because the middle point is where these molecules all gets recruited to. Because they're proximal there, they can actually interact to lead to downstream signaling. And then this structure plays a lot of different functions. And this signaling platform is what I mentioned already. But we also found we found that it's also important for axon-axon and axon-dendrite interactions. And we and others have found that they're important for axon and dendrite diameter control. And we and others have found that it is important for axon degeneration. And then it's also, we hypothesize it's actually important for the survival of axon under mechanical stress. And another lab, Matthew Raspin lab, actually knocked out and perturbed the structure and found that it's actually medically important because once you perturbed it, you can actually lead to neurological disorders in mice. And more recently, people found that structure perturbation of this in humans through natural mutation actually cause diseases and so on. So this is just a structure that we discovered by super resolution imaging, and now we know they're actually medically important. And then I end this part by showing you that, originally we thought that this is a very stable structure because it is important for the axon survival under mechanical stress. It's important as a signaling platform, so it should be constantly there. Not only so, when we try to disrupt that with drugs, it was very hard to destroy that. But it's only when we actually imaged it live in real time. And then I'm glad that this movie plays, and then you can see, seeing is believing. It's highly dynamic. Locally, it keeps dissolving and reforming, dissolving and reforming. And then the postdocs spent years to figure out the molecular mechanism, which I won't belabor you with all the experimental results, but just to tell you that exactly that's actually not a spontaneous thing. It's actually enzymatically driven, multiple enzymatic processes that are calcium influx induced that can actually have phosphorylation and protease activity that destroy the ring and make them locally unstable and then reform and so on. So that it's highly dynamic, surprisingly. And then you might wonder why, as I said, I thought the structure is so stable. Why is it dynamic? Usually biology doesn't do, if it's spontaneous, you could say that, okay, it's just spontaneously unstable. But if it takes so many enzymatic processes to make it dynamic and spending that much energy, usually it's for a function. And we think it has probably other functions, but at least we found one reason why it is dynamic. And like I said, it's a functional important molecule itself. So it needs to be there. But on the other hand, it's a dense matrix right underneath the membrane of the axons and dendrites and so on. So because of that, it also prevents other important processes from happening. For example, one process that requires the bending of the membrane to form endocytic vesicles. And if you don't know what that is a process for the cell to take up things. Often people call it a cell-eating process. Now, that is actually inhibited by this mechanical structure underneath that. So in order to have the structure to perform its own function, yet not preventing some other major functions that happening at the membrane, and then we think that the cell invented this process, invented this mechanism that make the structure locally unstable so that these other processes can also happen, but this structure is also for the most time there. And then I didn't show you the results. Indeed, when we actually, now that because we understand how enzymatically to, the mechanisms of making, the mechanism underlying the dynamics, we can make it not dynamic. And when we make it not dynamic, these very important endocytosis processes were strongly inhibited. So cell is a very smart thing. It can invent, if I think about it, it's so smart that I have a structure, I need it. I also don't want it to prevent other processes, so I make the structure locally and temporally dynamic so that I can allow both kinds of functions to happen. So with that, I would like to say that, not only us, but other labs, so like I mentioned, Stefan Hell's lab, Eric Beswick's lab, other labs, not only like microscopy, but also electron tomography, such as the beautiful work of Wolfgang Bollmeister and so on. So all this is so that we hope that eventually we have this dream that will become true, that is, you can really see what happened in the living systems at the molecular level, and then so that learning the molecular mechanisms of life processes, how it goes wrong in diseases. Movie playing is really not working that well tonight. So it's just like a fun process of watching movie, and while you're doing movie watching, you're learning important knowledge. So with that, I should say that if this is already a real movie, then a lot of work is already done. So this is obviously not real, so it's artistic rendering. So with that, let me thank my people who are doing this work. And again, if STORM is also decades of work, so because of that, if I actually acknowledge all of them, I would take too much of your time. But I would still emphasize that we professors enjoy directing people, making talks, and so on. But the hardworking students are the true heroes. And over the years, I have had the fortune to have many talented students. And they made my life a lot more fun than otherwise it would be. And then the people's work who I talked about are underlined. And then you can see many of them are actually having their own very successful labs and so on. So with that, let me thank you for your attention and close this talk.
Thank you, Professor Zhang, for your wonderful talk. So your work shows us how advanced imaging can reveal life at the smallest scales and open new paths to biological discoveries. So I think due to time limit, we may have the time for 1 or 2 quick questions, do we? Maybe we can leave all the discussions during the dinner time. Thank you very much, Professor Zhang. Thank you.
Thank you.
Yeah. All right. Thank you again. So next, we will welcome Dr. Xiaodong He, Senior Vice President of Jingdong Group and Head of AI Innovation. Dr. He is a leading expert in artificial intelligence. His work covers natural language processing, multimodal AI, large models, and AI systems. Before joining Jingdong, he worked for many years at Microsoft Research. At Jingdong, he has helped bring AI into real-world applications in retail, services, industry, and in particular, intelligent interaction. So his talk is titled From Digital Human to Humanoids. So please welcome Dr. He. Welcome.
So thanks Professor Low's invitation. It's a great honor for me to come here and have this opportunity to share some of our thoughts on AI, particularly on multimodal AI and how it's going from the digital world to the physical world. And we start to build a lot of applications. Eventually, we hope people will love the application, love the products, and from this application and products to feel the beauty of AI. So before I go to the details of technology, actually, I'll show how actually public feel the AI. This is a picture I took last year at the World Robot Conference at Beijing, and I'm actually really impressed and touched by how regular people feel, interact, and play with AI. So you can see a lot of kids and the parents love it. Yeah, and like the dance robots as well. Yeah. But we know actually the technology behind these robots actually has gone a long way. Now we actually can think of back to like originally we start from the perception intelligence, like speech recognition, like face recognition, like image recognition. That is some kind of intelligence that actually animal also have. We call it perception intelligence. Then we move to, we call it cognition intelligence, like language understanding, like knowledge, like reasoning, like conversation. Then moving forward, actually, we don't limit ourselves to only one modality. We move to multimodal intelligence. We combine information from image, from language, from sound, from all together, and start to build more interesting applications, as I will show later, like digital human. And then moving from the digital world to the physical world, we apply this multimodal intelligence to the machines, to the robots, and make a robot have the ability to interact with us, to work for us. Then we start to get this embodied AI, and then we eventually build these robots that can actually work and even live together with us. So there's a long way to go actually from technology point of view. Back to the history, you can see this wave of AI, actually the fundamental technology is deep neural network, which have been going actually a long way. And one of the breakthrough have been made by Geoff Hinton and his colleague is in like 2006. He published one of the landmarking paper in Science and start to see how we can push this shallow layer neural network to deeper. And once it's getting deeper, actually things emerges like much more powerful capacity, make the AI system start to perform much better on some core tasks. But we don't stop at a library or at a university. In 2006, they published a paper, and in 2008, actually at that time I was still in Microsoft, and my colleagues at Microsoft invited Hinton to our workshop. And we had to think of, can we actually move this new deep technology or deep neural network technology to apply on some key applications like speech recognition, which is still an open question at that time. And this is actually how speech recognition goes is as a task. So at the early time, I mean, this is a very challenging switchboard speech recognition task has been going through for many years, originally initialized by DARPA. So at the beginning, as you can see in the red curve, the speech recognition error is very high. Almost at the beginning is as high as almost like 100%. I mean, basically AI can do nothing about speech recognition. Then going through a long way, people develop like hidden Markov model and the Gaussian mixture model, this statistical model, to bring down the speech recognition error rate to like 25 or something like that in the year of 2000. But then the whole progress stopped for almost a decade. In this decade, people tried like discriminative training, like large margin training, and only get small improvements. Then in 2012, as Hinton worked with us at Microsoft, we started to see another breakthrough. The error rate started going down again, which is the yellow curve. And after several more years, in like 2017, eventually Microsoft declared we are reaching the human parity. Basically, AI perform speech recognition as well as a professional human transcriptor. But this is a success, but only success in the perception intelligence area. The language is another story, which could be even more difficult. And as a little later than speech recognition, people start to work on language understanding using deep network. Like the pioneers like Thomas Mikloff, like Yoshua Bengio and their colleagues, they are thinking how we can push neural network to perform on language understanding kind of task. Many people don't believe that at that time because language is naturally a symbolic signal. It's unlike the speech or even image, which are continuous signal, which people think neural network naturally shall perform well. But the language itself is like symbolic, it's like a symbol one by one. So one of the, I would say, a brilliant idea proposed by Thomas Mikloff is the word embedding, to use a vector to represent, to use a continuous vector to represent the meaning, the semantic meaning of a word in the language space. That actually bring neural network into the language modeling space and start to take off. And actually, since many people don't believe that in that time, Actually, Thomas Mikolov later, when he received the Test of Time award, he comments that his paper is almost rejected actually many times, eventually got into this one. And nobody believed this is going to work, but after 10 years, actually people see this is actually a breakthrough and award him a Test of Time award. Similar happens to some other work we have done. For example, another work we have done with at Microsoft is use deep learning to do semantic understanding as well. So at that time, we think like 10,000 cores and like 10 billion of text is large scale at that time, which now we think is only a small scale. We are actually over 10 years, we are like 10,000 times more scaled up compared to that time. And everything changed once you scaled up. And so what I want to say is that, today we talk about big model, we talk about AGI, but this is not just like one shot technology. It's actually a decade-long scientific innovation and engineering scale up all together, and all the institutes around the world work together. Eventually, we start from water embedding to attention to transformer, and eventually we get to the GPT area. And then the similar thing happens in the multimodal, in the image area as well. We start with the GAN, then go to attention, and eventually we go to the diffusion and diffusion-based transformer, and to nowadays Sora and even CDANs. So over this long run of technology journey, I will say a lot of institutes, including our JD, actually make our contribution to make this work. So now people actually truly believe AI actually is there, and the large model is really powerful. But people may think why AI is, or why this deep learning driven large model actually works. This is an interesting analogical comparison between human brain and the large model, actually presented by Professor Hinton last year at Shanghai, and I borrowed his idea and present here again. Basically, it's a little like the human brain. The basic computation unit in large model is very simple. It's like a weighted sum plus a nonlinear transformation. Weighted sum and a nonlinear transformation to output to the label neural nets. In digital neural network, that was the new digital neural network. And it actually works similar as the human neural cell. But this small, simple computation combined with a large scale of numbers of this computation together make a big difference. If you put like hundreds of billions of this computation unit together, actually then the intelligence emerged. This is also kind of compared to like how humans have our own intelligence come up. We know nowadays we know actually from this different with combination of all the cells work together. Now different region in our brain actually start to emerge the intelligence in different areas, like speech intelligence or language intelligence or the vision understanding abilities. This is how actually we see in AI as well. So at the beginning, we see the AI start to work as different individual areas, like in speech recognition or like in the face recognition. But nowadays, if we combine this model even larger, if we combine this ability together and scale up the model even larger, nowadays, actually, we can see AI go beyond this basic ability, but going into this ability that we usually measure in how we measure the human. Like if we measure this human is smart enough, you can say, can this human be a salesman? We actually now ask the same question to AI, to judge the AI, if this AI is smart enough to be a salesman, or if the AI is smart enough to be a programmer, or if the AI is smart enough, even can perform fundamental research. And that's how we are right now. Yeah. So going beyond language, speech, and then we go to the multimodal intelligence. This is one of the things actually we built the product to the public to show the multimodal intelligence. Basically, that was 2016. Satya published one of the products we built at that time. At that time, you take a picture of anything. Then our AI will recognize and describe user natural language. This is the first time the multimodal intelligence to across two modalities. It's not just a recognition of a human in the picture, but rather than actually it's describe what is happening in the scene. And this is actually going further and become a product, and actually people can use it. For example, we even show this to to Jensen Huang at one time when he questioned if this actually work or not. And we also go to the San Francisco Zoom and to test in a real scenario to see if this actually is AI is really smart enough, can go beyond the laboratory and go into the wild world. And also another direction is, can we at that time ask AI to start to create something, like create a picture or create some art or some like vision content. It's another work also happens in like 2018. We ask AI not just describe what is happening in the picture, but we ask AI to draw the picture given a language description. So this is one of the birds we draw like in 2018. And interesting enough, at that time, when the reporter asked us what we can imagine, what is going to happen beyond that, we not brave enough. We always think maybe in one day, if we have enough computation, we can make the whole film rather than just make a single picture by AI. At that time, we think this is maybe impossible, but just an imagination. But actually, the technology moved faster than we thought. Nowadays, people actually start to use AI to create a movie. I saw some news, and I also have some friends in the Chinese Academy of Fine Arts, and they are actually creating a whole movie using AI this year. So in less than 10 years, actually, AI moving so fast. And also, as we can see, AI not just work on this content. AI actually move also to medical, to climate, to biological, and to material, all kinds of these areas. AI start to do this this research beyond just do some basic work. And also another thing, we thanks to the engineering effort, now thanks to the scaling up, now the cost of AI is also drop down quickly. This is like a curve to show in the x-axis is like the unit, the cost of performing a task, and the y-axis is like the accuracy or the how difficult the difficulty of a task that AI can solve. You can see the whole thing go up quickly, and eventually the unit price of using AI to solve really challenging tasks is going down quickly. That enable more opportunity to actually to develop more useful applications. And this one is another show that AI actually get into the content generation industry. And not just generate content for a single modality, not just generate content for sound or for text only, but actually they combine together. One of the representative product is like a digital human, as I show here. And I will skip a little bit of the technology details, but show the example. So this is an example we can see now. AI start to generate the content in a multimodal way. And more interestingly, we start to generate a digital avatar for real humans. This is an example in the right-hand side. I'll show you, let me show the example directly. So this is a digital human we create. So actually, this is our founder, Genesis founder, Richard Liu. But what you have seen is not actually the real Richard Liu. It's actually a digital avatar of him. So we create a digital avatar of our founder, and we put this digital avatar online to our JD.com and ask this avatar to perform this live stream sales. And in less than 1 hour, actually, he make a sales like 5,000 million. Yeah, 5,000 million yuan sales like in less than 1 hour. And in just like 1 hour, more than 10 million, actually 40 million views about this performance. Because in this case, we want to basically test. It's basically like a Turing test, but we will test if AI's generated content can be as good that people cannot tell it's from AI and actually can embrace this kind of content. And our results show actually nowadays public start to really embrace this kind of content and don't tell the difference and really accept this kind of task. And a little bit of technology details is like now, rather than use like more than 100 of the camera to capture the motion of the human to create the avatar, we only use 1 or 2 camera. Now, as less than like 10,000 yuan of cost, we can create such an avatar. And this is just like also goes through some process. At the beginning, we start to do this work. We in the research in the laboratory. This is how it's performed in the laboratory. So this is actually what we have done in 2023. That's the very first version of performance digital human. Then the second version is like this. This is famous female Dong Mingzhu, and we create a digital avatar for her as well. Yeah, so we now can start to use this technology to create this avatar for all this business person. So he or she can talk to their customer directly using the avatar. And then even later, we create a new generation of avatar that without using a model. So as before, we still need a human as a model to capture his behavior to create an avatar. But nowadays, we can create this AI native digital human without using any models. We can like this one. So every single pixel you have seen here, every sound, every text actually are generated out of nowhere by AI nowadays. So this is another view from us. Like we don't just live in the digital world. Although many of the content I show so far are in the digital world. Actually, we also have a lot of activity in the physical world, and also, of course, interact with our human bodies as well. So how we can move from the digital world to the physical world is our next step. For example, this is our overview how to perform this embodied AI. Probably due to time limit, I will skip the technical details. And this is not the research we're doing, but also I will skip the technical details. And this is some of the actual products and applications we're doing now. In the left-hand side is the robotic arm, which is deployed in JD Storm right now. Actually, we're using it every day. At the right-hand side is our latest version of the embodied AI or robots we're building in our laboratory. I will give an example. So in this task, we don't just ask the AI to do one task. We actually, you can see we have a lot of interaction in between. Yeah, this is another scenario at JD. We try to pack the Shenzhen, this fresh foods. Yeah. So not just stuff in laboratory. If we want to go beyond laboratory to really to the physical world to work in our scenario, we need a lot of data. So we also initiate another large-scale embodied AI data collection initiative. We are going to collect more than 1 million hours of the ego view data. Ask the real human to record their activity, like in this year, and hopefully we can scale up to like 10 million of hours of this data to enable the whole society to build the largest model for embodied AI. This also we propose this Joint Insight program to collaborate with all the partners. In GD, we provide the brain, basically is an embodied brain, and our partner can provide this the machinery, the body. So work together to speed up the development of embodied AI. And at the end, I will elaborate a little bit to show an example how we can collaborate with our partner, not just the robots, but all kinds of hardwares that work together to make the whole physical world intelligent. This is our vision. Yeah, so from digital human to humanoids, we hope we use AI to make our life better. Yeah, thanks. Yeah.
Thank you very much, Dr. He. Thank you very much. Well, from digital humans to humanoids, we see AI moving from the virtual world to the physical world with many new opportunities ahead. So I think, do we have any quick questions? If not, we will leave all the questions to the dinner time. Okay, thank you very much, Dr. He. Thank you. Now, please allow me to welcome Professor Julie Fegan, Distinguished Professor of Biochemistry at UCLA. Dr. Fegan is a leading structural biologist. Her research focuses on structure and function of nucleic acids, including G chondroplexes, rainbow nucleoprotein complexes, and also telomerase. So her work helps us understand how molecular machines in life are built and how they work. Her talk is titled A Scientific Lifetime Affair with Nucleic Acids from G chondroplexes to Telomerase. So please join me in welcoming Professor Fegan. All right, the floor is yours.
Can we plug it in? Okay, so I'm actually going to tell you about telomerase, mostly about telomerase today. And since I'm a structural biologist, I just want to introduce very briefly the techniques that my lab uses, which are NMR spectroscopy combined with cryo-electron microscopy. Primarily, we do a little bit of X-ray crystallography. And what's very important if you really want to learn about biological molecules and complexes is to make sure that you understand the biochemistry and molecular and cell biology. So I actually got interested in telomerase more than 30 years ago when my lab was doing NMR spectroscopy on unusual DNA and RNA structures. And we solved this structure of what turned out to be a quadruplex formed by the telomere DNA repeat from oxytricha, which is 4 T's, 4 G's. And so if you have a bunch of them, it forms this really amazing structure that was had some completely unexpected features, including the orientation of the bases in these quartets. We were able to establish how they pair, and also these diagonal loops. So this was at the beginning of when you could do NMR spectroscopy and solve structures, and this was the first structure we solved where we didn't know what it was going to look like before we got it. Now, since then, this has become an enormous field in which there have been numerous roles proposed for both DNA and RNA G quadruplexes in cancer, in disease, and in gene regulation. Basically, it ranges from amyloid to aptamers to SARS-CoV-2 RNA, and also they are used in nanotechnology. Okay, so that's where we started. And then for many years, I tried to get people in my lab interested in telomerase long before it was really possible to do anything. But let me introduce to you the textbook picture of what telomerase is. So a telomerase basically extends the 3' ends of linear chromosomes, which have the single strand overhang. So those 3' ends are mostly double-stranded with this 3' overhang of a G-rich repeat in one strand. This repeat is T2G4. It's found in Oxytricha nova. And then the other strand is the complementary C strand. And basically, if you read the textbook, telomerase is telomerase reverse transcriptase, or TERT, and an integral telomerase RNA TER, but really all you see in the textbook is this template. So basically, it's a reverse transcriptase. It gets recruited to the end of the telomere, pairs with this template, and then synthesizes a single telomere repeat, one, and then processively synthesizes multiple telomere repeats. So the template has to translocate and then make another telomere repeat. And at some point, this process stops, and another enzyme, which is the other half of the story, called DNA polymerase alpha primase, is recruited, puts down an RNA primer, and fills in the C strand. And all of this has to happen in the context of proteins that coat the telomeres in order to prevent the ends of our chromosomes from being misrecognized as double-strand breaks and wreaking all kind of havoc in our cells. So I'm going to talk about tetrahymenotelomerase today and human telomerase today. Tetrahymena is this beautiful little ciliated protozoan, and it's the organism in which both telomeres and telomerase were first discovered. And I just want to show you one kind of typical assay that we do to test the activity of telomerase. So you can see one telomere repeat, 2, 3, 4, 5. At the end of each synthesis, you get another dark band. And if there's a mutation that affects activity, you get less, you see less dark bands. So why do we care about telomerase? Telomerase is of enormous medical interest because it is a highly regulated determinant of cellular aging stem cell renewal, and tumorigenesis. So I just told you how important telomerase is, so you would think it's always active in the cell, but contrary to that, most somatic cells have no detectable level of telomerase activity. So here's a nice young telomere or chromosome with long telomeres, but this old chromosome has short telomeres, and that's because our telomeres shorten with age. You don't want your cells to be able to replicate forever. So at some point you get, when telomeres reach a certain point, the cells go into senescence or apoptosis. However, telomerase is upregulated in almost all cancers. In fact, mutations in the promoter for TERT that upregulate its production are one of the most common mutations in cancer. And this upregulation is important for the immortal phenotype of cancer cells. So there are also a huge family of what are called telomere biology disorders. Those arise from mutations either in telomerase, including the telomerase RNA components, or telomere components, and they're linked to a variety of both uncommon diseases like dyskeratosis congenita, but also things like aplastic anemia and pulmonary fibrosis. And in those cases, people who inherit these mutations actually have short telomeres. So on the one hand, you might want to increase the length of your telomeres if you have these diseases, but prevent them from being activated if you don't want to get cancer. So it's a complicated system that I don't have time to go into much detail about today. Okay, so this is a slide from a review we wrote 20 years ago when people were just starting to figure out what does the telomerase RNA look like. So what I showed you in the previous slide is this template, but there's all this telomerase RNA. And this was the goal of my life at the beginning when we first started working on this, was figuring out what does the rest of that RNA do in this beautiful enzyme. And so one of the things you can see is all of these different organisms, they have different lengths of telomerase RNA, but they have some kind of conserved features, and one is this large or small pseudoknot, and another is a region that's called a stem terminus element that I won't talk too much about. And so we started by looking at these various pseudoknots, especially the conserved part of these pseudoknots. So all of these are various papers where we studied really just the RNA, mostly by NMR. And this was the first important structure that we solved. This is the conserved part of the telomerase pseudoknot in human. And we got the cover of Molecular Cell because after I talked about this at a meeting, the editor of Cell kind of wanted to apologize to me for not accepting our paper to Cell and sending it instead to Molecular Cell because she saw the reaction from the audience. So this turned out to be a surprisingly important paper because we discovered that in these pseudoknots, which I guess I should have pointed out for those of you, is there's like a loop paired to a stem and another loop paired to another stem. So it's not a true knot. But what was surprising was to find that the loops paired with the stem to make these base triples. So this would be UAU, okay? And this was the first time that anybody had seen that you could form base triples in the major groove of RNA. For some reason, RNA biologists thought this wasn't possible because the groove was too narrow, but in fact, it's quite common. And so this paper keeps getting cited for that reason, even to this day. Okay, and then we could correlate the formation of those base pairs to function. So if you mutate them, bad things happen. So there are This slide is just to give you an overview of what we really know now. So there are 4 key players in human telomeric DNA synthesis. So I told you there's all these proteins on the telomeres. There's a 6-protein complex identified by Tizia Delanga. This is just one of those 6. It looks like very structured, but it's not. And there's various combinations of them. So this is called shelterin in humans. And the only 2 proteins you need to care about for this talk are POT1, which binds the single-strand overhang, and TPP1, which interacts with telomerase. So this is our planet telomerase, picture of telomerase. These are the components that have been established and seen in cryo-EM structures from my lab, Kelly Nguyen's lab, and Ming Lei's lab. So I told you at some point telomerase stops. It's inhibited by this complex called CST, and CST recruits that other polymerase, pol-alpha primase, to synthesize the C strand. Well, when we started working on getting the whole structure of telomerase, we started working on tetrahymenotelomerase because at that time it was the only telomerase for which all of the components were thought to be known. And so this is what tetrahymenotelomerase looks like, and it looks very different. But it turns out that there's all these proteins in tetrahymenotelomerase that actually correspond to proteins that are constitutively associated with tetrahymenotelomerase, but they correspond to proteins or are homologous to proteins that only transiently associate with human telomerase at telomeres. So this TEB1 is equivalent of POT1, and this P50 is equivalent of TPP1, and this 3-protein complex, which was just named by numbers, molecular weight, it turns out to be homologous to this CST complex, and then tetrahymena has its own pol-alpha primase. Okay, so when we started, it was back in the dark ages of EM. We actually started, people were doing cryo-EM, but we started with negative stain EM. So we got this 25 angstrom structure, which might look like something only a mother could love. But in fact, it was important enough that it got accepted into Nature because we were able to locate all of the components and also say something about we did a little bit of modeling. There was a whole bunch of really cool experiments that went into getting this structure and also other things we learned in the process. Okay, so then it took us another 2 years to get to 9 angstrom structure. In cryo-EM at 9 angstroms, you can start to see alpha helices. And so if you have any models of proteins or homologs of those proteins, you can start to fit them in. We had a crystal structure of this protein P19, which turns out to be part of the CST complex. We had models for some of the reverse transcriptase and some of this protein P65 where we had a crystal structure from my lab. And another thing we learned from this structure is that we actually discovered that there were 2 additional subunits that had not been previously identified. This turns out to be common when you do electron microscopy, is that you discover new components that somehow didn't show up in the mass spec or were missed. So we confirmed them by mass spec. And then finally, we got to 4.8 angstroms. Here, the CST complex is masked out. And at this resolution, we could just barely make a model if we wanted to kill ourselves for a year. During the same year, we tried to get to higher resolution. So that's where we were in 2018. And then there was the pandemic, and we had a lot of data, and we managed to get a lot of things done. I'm not going to be able to talk about most of this, but I just wanted to show you these highlights. So we got to 3.3 angstroms resolution. We also got several different structures, and I'm going to talk about this in terms of what we learned about mechanism of synthesis of the G strand. we also solved the structure of human telomerase. So we could compare what we learned about mechanism for tetrahymena to mechanism in human telomerase. And we also had this protein TPP1 that recruits telomerase to telomeres in our complex. So we learned something about recruitment. This complex, we finally solved this dynamic 3-protein complex CST, both by itself, as shown here, but also in a complex with polymerase alpha primase, so we could see a little bit about how once a G strand is synthesized, it's used as a template for the C strand. And finally, we also solved the structure of this assembly biogenesis protein P65, and the reason I want to emphasize this is that we could only do this by combining cryo-EM and NMR spectroscopy, because this is a very dynamic protein with different domains connected by flexible linkers. And so I'm going to talk about this and this, and at the very end, I'm going to tell you about some very new work on a structure of a telomerase inhibitor bound to telomerase. Hopefully, I have time. Okay, so here is the 3.3 angstrom resolution structure of telomerase with this dynamic CST complex masked out. This, I just love to show this. These are actually just a bunch of 2D reconstructions from the negative stain, and I show it because you can see how dynamic this RNA protein complex is, and I'll turn it off so you don't, start having fits. And so before I show you the movie, I just want to point out that whenever you do cryo-EM, you always have you have this beautiful structure. It looks like you have everything, but everything that's in gray here, either dark gray or light gray, are regions that we never see in the cryo-EM, either because the protein is unstructured or because it's there's a domain connected by flexible linker. Okay, so this is the TERT in blue. I'll show you the different colors in a minute. And this is all color-coded. So here's P65 down here. That's the map I showed you. This is the model. Fit into that nice, beautiful map. And now this is the TERT by itself. And now you can see the different domains of the TERT. So this is the RNA binding domain, the reverse transcriptase domain, and the CTE. And then here's a couple other domains. I'm going to point them out in a second. And one of the big surprises in this structure, because we kept expecting to get a long DNA RNA duplex as we made essentially made the DNA, we only see 4 base pairs. And I'll show you why that's significant in a minute. Okay, so here you can see the structure of the TERT. So it has this TERT ring that's the RNA binding domain, the RT domain, and the CTE. And if you know anything about polymerases from your undergraduate biochemistry course, the reverse transcriptase is the palm and fingers, the CTE domain is the thumb. And then we discovered this unexpected extension in the middle of the RT that makes this complex with this telomerase essential and terminal domain. And this turns out to be really important for function. This complex was completely unexpected. And so now you can, I'll show you how the RNA wraps around the TERT. So we're going to start here at the stem 1, and then here you have this, right here, this stem loop 2, and then you have this single strand RNA that's running across the front. And then in the back is the pseudoknot. Okay, that's this part. So we just went around this circle. I talked a little bit too slow, believe it or not. And then here's this region where this end of this stem loop, this loop inserts between the CTE and the RBD to close the TERT ring. Okay, so in the front, and I'll show you this in the next slide, there is just kind of this single strand region that includes the template. Not in the next slide, the slide after this. So we had this high resolution structure. We actually had 4 of them. So the question is, can we answer the question, can we say something about the structural basis of individual, the telomerase nucleotide addition, and also its processivity? So this is a little movie that we had made. So the DNA primer binds, we synthesize our 6 nucleotide telomere repeat, and then somehow the template resets, and we synthesize another telomere repeat. This is the structure I just showed you with the DNA coming out here. And I told you, so we actually saw 4 structures. This is the one I just showed you, and there are 4 base pairs. 2 of them are in the alignment region. 2 of them would be 2 new nucleotides being made. And for every step, it's 4 base pairs. Now, you are seeing 5, and that is because it hasn't yet moved out of the active site. We used these modified oligonucleotides, and so it's a little bit more too stable, and it doesn't move out. But initially, once this base pair forms, it moves out. So we have step 2, step 3, step 4, and step 5. And fortunately, the 2 highest resolution structures are the farthest apart. And so we could say something about how the RNA moves through the enzyme as synthesis proceeds. And we also saw all kinds of different important interactions that we could monitor, including this thing that we call the bridge loop at the top and bottom of the duplex. Okay, so here we are looking into the catalytic core, and we have a region in pink that is locked onto the protein. And then we have these linkers. Here's our template, and on either side are the linkers. And so the linkers are moving through in it's kind of an accordion, right? So this stretches and this contracts as it goes through. And so that's how the template can move and processively move the nucleotide position into the active site. So this leads to a model for telomere repeat synthesis that we made. So the top half is adding the 6 nucleotides. The bottom, and this is pretty solid. The bottom half is how then the duplex comes apart and so that the template can reset. So I'm just going to do a quick step through. So initially, the DNA aligns and pairs with the template alignment region, and then steps through adding nucleotides. So there's 6 steps of nucleotide addition. So I just want you to remember the alignment part. And then when you get here, the next important thing for you to remember is this, is that once you get to the 6 base pairs, we started thinking about what was in the literature. And so there was this really beautiful work from Pei Guang's lab in which they had seen and proposed that the product strand, or the DNA primer in this case, starts to move and then the template follows. But in the case of telomerase, the template cannot follow because it's anchored here at this point. It's stretched as far as it can. And so that destabilizes those 4 base pairs so they can start to open up. And then the template can move back to the beginning and can close up, repair, and start the cycle again. And so instead of, I had been trying to figure out how do you melt that duplex so you can do each successive round of telomere repeat synthesis? But it's exactly the opposite problem. How do you hold it stable? How does it stay stable enough and paired through the synthesis? So there has to be the enzyme holding various interactions with the amino acids on the enzyme holding it in. Okay, so as I mentioned, we solved the structure of human telomerase. So this is the TERT. And then we also have this TPP1 that we added that tells us about recruitment. I'm not going to talk about this biogenesis domain down here. It's something called an HACA RMP. All the proteins from HACA RMP that have been co-opted into human telomerase, for those who know some biology. And then this is the catalytic core, and here's the TPP1. One big surprise that was actually discovered by Kelly Nguyen's lab, not by our lab, is that two histones, H2A, H2B, also appear to have a role similar to P65 in tetrahymena. And we were able to model the RNA with the help of our NMR structures of the entire pseudoknot region. Okay, so how does human telomerase catalytic core compare with tetrahymena? And I think you can see that they are pretty similar. Now, there are some significant differences, but here you can see that what we learned from tetrahymena translates, at least in the front, how the template moves through the active site pretty clearly. And but a big difference is that there's this long double-strand region in the pseudoknot that actually bumps up against this 10 domain. And here, this is just to show how clear the density is for those 4 base pairs. And these are some activity assays that we did that show the importance of those 2 bridge loop amino acids, where if we change them to alanine, there's almost no activity, so this lysine to an alanine, but if we change it, for instance, to an arginine, which is what's found in tetrahymena, the activity is pretty much the same. So that's just an example of the kinds of ways that we check are our conclusions about activity actually borne out by the biology. So we were also able to compare how P50 versus TPP1 interact with TERT. And I think because I want to get to the next part, I'm going to skip over this. But we were able to see that they're very similar in both tetrahymena and human. It's a kind of cool story, but there's a few other things I want to tell you before my time runs out. So, you know, the great thing about having a structure of human telomerase is then you can start thinking about, can we do anything about disease? There are like database of like over 100 different mutations that have been discovered that cause these telomere biology disorders, and many of them are in TERT and TER, and that's what's mapped here. Are all of the mutations that are known that are in TERT and are in TER, and one of the amazing things is how many of them are at the interface of the telomerase RNA and the TERT. And so we actually looked at every single one of them, and we can say how they disrupt interactions. So this is kind of a beginning window into maybe someday being able to think about what we can do about disease. So in my last few minutes, I want to tell you about our work on telomerase inhibitors that was just published. So this is from a review that was recently published. And basically, people have been looking at telomerase inhibitors for 25 years with very little success at developing a drug. So there's now 6-thiodG is in phase 3 clinical trial against non-small cell lung cancer. And this one, developed by Geron, which is basically about a 16 nucleotide oligonucleotide that's complementary to the template in adjacent region, has been approved for a small subset of myelodysplastic syndromes. It's called imetelstat. It's basically an antisense oligonucleotide. The one we decided to look at is this one, BIBR 1532, which was looked at by Boehringer Ingelheim 25 years ago. And so here is its IC50, which is pretty good, but it hasn't progressed anywhere to a clinic because of low bioavailability and IC50 when you go from in vitro to in vivo. However, it is used widely in studies of telomerase and telomere function. And there was a previous structure from more than a decade ago from this very primitive flower beetle TERT that's missing the 10 domain, missing the trap domain, and in fact for which no telomerase RNA had been found until about last year. And that if it's active as a telomerase, it's not very active. Nevertheless, Emanuel Scortolacchi solved the structure of this primitive TERT in complex with this BIBR1532 by X-ray crystallography, and here you can see it in this binding pocket on the CTE domain. So when we solved the structure of human telomerase, we thought, oh, well, if we look at that same region on human telomerase, the telomerase RNA is there, and this U177 and this U307 are both critical nucleotides. If you mutate this, you really disrupt activity. And so, and if we fit the BIBR1532 into that site, you can see it looks like it would disrupt a really critical TERT-TER interaction. So we might have this, we thought, oh, that's how it's working as an allosteric inhibitor. Somehow it's doing something to the RNA interaction with the TERT. So Yuqing Wang in my lab solved the structure. Here it is. And when he looked in that region, he was very disappointed for about 24 hours because there is no BIBR in the expected binding site. However, instead, well, basically he went home all depressed and came back and decided, I'm going to look elsewhere. So where is it? It's actually in the RT domain, not too far from the template, but more than 12 angstroms from the active site. So here it is. There's a series of beta strands around it. It's right here. And these 3 red balls indicate the active site. They are catalytic aspartates that are found in all polymerases. Okay? And this is what it looks like. The interactions are completely different. The orientations are completely different. This is what medicinal chemists, how they draw interactions with a drug. So we know now why every effort to improve this inhibitor failed, because they were using a model that was incorrect. But when we looked at what does this drug do in its binding pocket, it has this very nice binding pocket. It barely affects its structure at all. So you can see this is just the backbone, the backbone of the protein. It barely moves. The biggest conformational change is this histidine moving from here to here, but really not very much going on. So how is this enzyme inhibiting? I mean, how is this small molecule inhibiting telomerase? So here it is here. And we did these activity assays where we mutated many of the residues in this binding pocket to alanines just to show how important this binding pocket is. I'm almost done. Okay. So Yuqing decided to look and see if he could figure out what was going on by looking at each step using an enzymatic assay in which he could individually look at addition of the nucleotide to each step, okay? And he knew from work of Julian Chen that in human telomerase, the first step of nucleotide addition is rate limiting. Okay, so he did this experiment in which he added dideoxynucleotides, the appropriate one. So it can add, but no additional nucleotide can add because the hydroxyl group that needs to be available for adding on an additional nucleotide is blocked by, is a deoxy. Okay, and then he calculated the inhibition effect relative to the enzyme without BIBR. And you can see that the first step is disproportionately inhibited by the enzyme. So he decided to obtain more structures for the alignment step and for the first step. So we went back to the drawing board. So for the alignment step, here we have the DNA aligned with the template, with the alignment region of the template. So you can see exactly what we expect to see. And when you have BIBR there, you still see the same thing. Doesn't affect alignment. So what's going on? So then he added a nucleotide analog that can go into the active site but cannot add. And so here it is sitting in the active site, exactly what we expected to see. We expect to go from here to here. But when he looked at the samples with BIBR, what he saw was that he could not fit a single, if he tried to calculate a single structure from the EM map, he couldn't get one. What he saw instead, he was able to sort them out into 3 separate structures. So this indicates that things have slowed down so much that you can see the intermediate steps in which the DNA product strand starts to move. Remember I told you, starts to separate to move to the next step. That is followed by the template. And then here, although you can't see it, there's only just a little bit of density, so the nucleotide, incoming nucleotide is kind of coming in and out, but it's moved to the next step. And then finally, here's the incoming nucleotide. So we can actually see from the EM that BIBR is slowing things down. And that's what's summarized here. Okay, so BIBR 1532 is slowing the nucleotide translocation step. All right, so I'm sorry I'm running over, but basically you have to have this template reset pair with it's contacted by the same amino acids at each step. There's this functional unit for nucleotide translocation. And so basically, you have to have this movement, these dynamic movements, and those dynamics are damped by this inhibitor. So that's summarized here. This is the important thing that you need to remember. And now we need to prove that this is correct. We also learned a lot about mechanism that I don't have time to talk about today. This is just a summary of all these different things that I mentioned today, but I don't want to stop without acknowledging the incredible people that have been involved in our work on telomerase from the very beginning to the EM. And this all started, the tetrahymena part started with a sabbatical in Kathy Collins' lab. All of the EM work has been done in collaboration with Hong Zhou, including joint postdoc Jensen Jiang, and more recently Yao He. So Yao did the tetrahymena telomerase mechanism stuff. He did CST pol-alpha primase. Yachang did P65. All of these guys have academic jobs. And Baocheng did human telomerase, and Yuqing did the work I just showed you about the inhibitor. And I'll stop there. And thank you for your attention.
Thank you very much, Professor Fagan. Well, after knowing, what you are doing, I think that's why, you look so young. Anyway, we have a very shy audience today, and also we have a very tight schedule. Thank you very much. Please be seated. Thank you. All right. So from the 3 speakers, we have learned the story from nanoscale imaging to embodied AI and humanoids, and then to nucleic acids, and also, the biological mechanisms, and also the talks have shown us how basic science and engineering can meet in powerful ways. They have also prepared us well for the discussion that follows. Okay. So the next part is the panel discussion on basic science in the AI era. And this panel actually follows the 3 remarkable keynote speaks. Professor Xiaowei Zhuang showed us how imaging can illuminate biology at the nanoscale. And Dr. Xiaodong He discussed the path from digital humans to humanoids. And Professor Julie Fagan took us through a scientific lifetime with nucleic acids. So together, these talks raise a broader question. As AI becomes increasingly powerful, how will it reshape the basic science? Will AI simply accelerate discovery? Or will it change the way we formulate questions, build theories, design experiments, and maybe validate knowledge? So now we are very honored to have 5 distinguished panelists with us today. So the first panelist is Professor Shitong Yao. So please join me in welcoming Professor Yao to the stage. Yeah, Professor Yao. And our second panelist is Professor Zhiquan Luo. Welcome on the stage. And our third panelist is Professor Robert Tarjan. Welcome. Our fourth panelist is Professor Jingchao Xu. Welcome. Last but not least, Professor Yonggang Huang. Welcome. So their expertise spans from mathematics, algorithms, optimization, scientific computing, mechanics, materials, and engineering sciences. So we look forward to a rich conversation across these fields. And because we have a very tight schedule, so I will directly short the questions. So we will begin with a very broad question to all the panelists. And Professor Yao actually mentioned it a little bit, in the welcoming speech. That is, in 1 or 2 minutes, how would you define basic science in the age of AI? Is AI merely a toolbox for accelerating science, or is it actually becoming a new way of doing science? So let me start from Professor Yao.
Well, whereas I see it, basic science is a science that are driven by curiosity and for truth and for its beauty. But AI certainly can help us to understand that. And as I mentioned, in the last few thousand years, each time when basic science was making a breakthrough, is based on some technological improvement, and it helps tremendously. But with the technology, we still have to think. Without deep thinking, nothing can be moved. And that's what I look at what is going on in AI. I think AI would help us to move up very fast, but it will reach a plateau. We need to have deep thinking. That's what happened. Yeah.
Thank you, Professor Yao. Professor Luo, do you want to have any, more explanation? Well, actually, they can share. Yeah.
I think AI is certainly real. It's very powerful. It's getting powerful as we speak. It's going to tackle many problems, surpasses human ability in many fronts. We have seen calculators surpassing human calculation capability and memory capability, face recognition, and so on. Now we're talking about inference capability. Some of these have already, many mathematical conjectures have been disproved by computers, by AI systems. People may be disappointed, but to me, I think we should not be disappointed, because having a counterexample review some structures maybe we have not seen. Maybe we should study those counterexamples and reformulate the conjecture. And then that's how we iterate and move the science forward. So I'm actually excited about the AI era, and then but we do have to be careful in how to use it, how to use it safely and then creatively and usefully, not to create risks unnecessary, that will disrupt the discovery process or to be detrimental to the humanity. I think that's very fundamental to what we'll be doing. Yeah.
Right, great. So Professor Tagen, what about you from a computer science perspective?
I think certainly AI is going to accelerate research. One of the things it's clearly good at is helping researchers to gather existing results, synthesize results, explain results. I think the interesting question is to what extent these systems can build theories, raise new questions. How creative can they actually be? I think it's an open question. I wouldn't care to speculate. Things are moving so fast, there's no way for anyone to predict what things are going to be like in 5 years, let alone 10 years. I think 1 year maybe we can extrapolate. I'm looking forward myself to experimenting with some of these systems. My younger colleagues already are doing this, but I think the real question is how creative can these systems be?
Thank you very much, Professor Xu.
I would offer some perspective from, I'm a computational mathematician. In the era of AI, I would not change the definition of what is science is. Scientific research is still, the basic objective is to make scientific discoveries. It's the how we get there that what changes by the AI. And for example, scientific computing in my area, we traditionally start from physics principle. We build mathematical models, and we develop a numerical method to solve it. With AI provides a complementary approach. We learn things from the data, and where maybe mathematical models is incomplete, or maybe too complicated, or we don't really know all the mechanism. Learning from the data can be complementary to what the traditional method would not be able to do. So by combining traditional method and the data-driven research, makes the scientific discovery and more efficient.
Thank you very much, Professor Hua.
I'll skip this question because it's on basic science. And my background is engineering. Engineering is not really basic science yet.
Okay. We will ask you more questions. All right. So my next question is actually to Professor Yao. I think nowadays, what people see is how AI is helping mathematics. Most people see, right? They are proving proofs, etc. And some of the proofs are being proved to be wrong. Some of them are being proved to be right. But I believe that AI also needs mathematics. So what kind of mathematics do you think AI needs in order to become a reliable engine of basic scientific discovery?
Well, there are many things that AI work out, like miracle, but we do not know why it works. And I think that's a very dangerous trend. Historically, in mathematics itself, that happens. About a century ago, the Italian algebraic geometers developed a lot of really beautiful and interesting statements about projective geometry. Most of them were right, but then they treat so-called genetic case, the case that happens most often, and they forget about the singular case, the case that does not happen that often. And then mistakes will start to accumulate. After, I think, in the order of 20 years, it got totally stuck and could not move anymore. So the whole algebraic geometry was reorganized in America instead of in Italy. and that's around 1930s, and it's completely reformed, and it becomes to move again. So I believe this is similar thing happens in AI. There's something that we always assume is true because the genetic case is true. But then you forget the most important singular case. And if you just forget once or twice, probably won't matter. But if you forget too many times, then it becomes a problem.
I see.
Yeah. And
Okay.
this one needs to deal with, I think. And because history told us that there was a problem. There are many other things that I think AI did not recognize. Too much linear approximation, and I think at the end it becomes the parameters is so huge. You need trillions and trillions of parameters. I think this should be cut down by understanding the linear, nonlinear problem directly instead of doing too many linear approximations. That's my view. Okay.
All right. Thank you very much, Professor Yao. And just to mention, a number of large-scale parameters, I guess I want to pose next question to Professor Luo. You're working on this optimization, and I believe optimization is at the heart of the current, AI. So from training large models to controlling robotics and also processing scientific data. So what are the most important optimization challenges behind the current AI systems, especially as AI moves from the digital environment to the physical world?
Thank you. That's a very important question. The current AI system is an engineered system, human-made system, to simulate how brain works. For that, you need a mathematical model that's basically a neural network. You need massive amount of data to simulate the learning process, and that learning process requires an algorithm to process the data and then to distill the information and then keep it in the network to produce intelligence, so to speak. So that process is all carried out on a supercomputer. Hundreds of thousands of GPU cards. And these are interconnected, and then lots of information passed back and forth. And they need to be processed quickly, and also with there's a lot of storage requirement. So the memory becomes a real bottleneck whenever you connect so many cards, so many computing units together. So the storage memory becomes a very important thing. So people are trying to cut down the memory, so to lower precision. So therefore, algorithms that can implement it in a parallel architecture, distributed fashion, asynchronously, and also with low bit precision. In that kind of environment, the traditional optimization algorithms, vast majority of them, unfortunately, don't work when you scale to that size, to that level. The only most successful example is the Nesterov, so accelerated gradient methods. That really is the bedrock of many of today's AI systems. It's implemented in every system. By the way, Yuri Nesterov is now full-time in Shenzhen, working in the SLAE. He's my colleague now. So now for this, I think we need to study the algorithms that can scale to that size with that limited precision. How do we make sure the error due to quantization, due to memory and so on, memory limitation, how to make sure they don't propagate. We need to study laws, how error gets propagated from layer to layer, from parts of the network to other parts. How do we make sure the algorithm actually stable, can stay stable? The other challenge when you come from large-scale system, I agree with Professor Yao. I mean, we need to cut down the size. Now we're going to trillions of parameters, and then many, many trillions. I think people are still scaling. Right now, training a large model, trillion-size model, the number I've seen is like $1 billion US cost to develop a model of that size. Now, going to physical world, like robots, we have seen the robots moving in real time and so on. That's another set of new challenges. You need multimodal things, for example, multiple sensors, very limited memory, real-time processing. You have to do real-time processing. Obstacle, I mean, you have to sense what is going on. You have to plan, long-range plan of you have a goal, then you have to break it down to stages and then make dynamic real-time decisions. And then control the robots and making sure they're stable and also accomplish the goal. So these present a host of new challenges. And then we can learn, for example, I mean, using a lot of data, I mean, creating data sets and so on. And that, I mean, Professor Dr. He mentioned already, this is facing the entire community is like really how to get useful data in an economic way and at scale so that they can be used to train robots, the intelligence robot. That's a hard problem. So many challenges for algorithm developers, optimization algorithm, real-time processing of information. Yeah.
All right, thank you very much, Professor Luo. So Professor Luo just mentioned that many traditional algorithms doesn't actually won't work, in the age of AI. But I want to pose a, counter question to Professor Ta Zhen. That is, from the perspective of algorithm and the data structures, just forget about the new algorithms. What are the timeless principles that AI researchers should not forget?
Before answering that question, I'd like to go back to the first question, which is, how is AI going to change what basic science is? I think we need to remember that basic science is a human endeavor, and it's humans who set the rules, decide what is important, what is beautiful, what is elegant. This is not going to change, and we should make sure that we're using these tools to serve us rather than some other goal. Now, coming back to the question, we know today that there is massive scaling up with hardware to the point where all these companies are building huge power plants to drive these computers. Efficiency is lacking here. The most important thing to me that they need to do is figure out how to build these systems in ways to really make them much more efficient. Algorithmic efficiency can give you nonlinear improvements in efficiency. Hardware can only give you linear improvements. I point out that this magic thing we have up here in our head, this is running on 10 watts of power or something like that. And think about what human beings can do with just a few examples. These systems, however magnificent they are, they've been trained on essentially the entirety of human verbal knowledge, large language models. So there's something that human beings still have the capability of doing that these large language models and many other AI systems, they're completely lacking. There are things we don't understand. So let us remember this point and let us strive for efficiency.
All right, thank you very much. So since the keywords, of your answer is efficiency, that moves directly to Professor Xu's, question. That is because I think the goal of scientific computing, one of the goals is really efficiency, right? So Professor Xu, so scientific computing traditionally begins with equations, physical principles, and also, of course, numerical methods. But machine learning actually often begins with data. So how should these two traditions be combined in the new age?
Thank you. Indeed, traditionally, scientific computing is mostly based on physics principle, mathematical models, and the numerical method. And in the age of AI, we are able to solve problems that may not be, we don't even know how to model the physics phenomena. So we're learning all the making discoveries, finding principles by studying this data. But I want to say that even for the traditional method, we can use the AI technology to make the numerical method more efficient. For example, we solve, say, Langevin-Stokes equation in fluid mechanics. We solve it very often. And through the machine learning, like operator learning these days, you can solve a whole bunch of Langevin-Stokes equation and offline. When next time you see a similar equation, you can use your train the solver to solve this equation much more efficiently. This would be much more efficient than by solving this equation from the scratch. And other things I like to also to say, the AI make it possible to make the maybe a sophisticated mathematical algorithm more practical. And indeed, over the years, for many mathematicians, computer scientists, we develop a beautiful algorithm. But sometimes these algorithms are very mathematically complicated. And mathematicians, we don't usually get our hands dirty to get the algorithm implemented for practical engineering applications. But engineers, practitioners may not have the expertise to understand the mathematics behind these algorithms. But AI, I believe, will change this. And AI seems to kind of expert for pretty much every subject that is involved. They know the mathematics, they know the physics. I just want to use the example, for example, like a multigrid method, which is something I studied for many years. And in the past, I was able, for example, to develop algorithm for, say, in Lawrence Livermore, they studied the fusion energy, simulation of the fusion energy, solving magnetic hydrodynamic system. In China, in PetroChina, we do the numerical simulation for petroleum engineering. And we were able to develop an algorithm that, say, is like 100 times faster than the traditional method. But unfortunately, this kind of efficiency cannot be easily realized for other applications. This is because it involves all the special properties of the physics of the numerical algorithm, and also all these ingredients the sophisticated algorithm would require. But AI seems to be able to do all these things. I'm really very excited in this area of AI, so that I may be able to use my multigrid method to solve most of the, if not all, the complicated engineering problems. One of such endeavors I'm taking right now, hopefully in Slade, and is to maybe develop efficient numerical method for simulations of the robot, like the performance we just see today, and through the physical laws and the mechanics. And I really very excited about the potential of the AI in the scientific computing area. Thank you.
All right, thank you very much, Professor Xu. So now, we have moved to the engineering and also physical world, so that I will pose the last question in this round to Professor Huang. So, Professor Huang, your work in solid mechanics and also flexible electronics connects the fundamental mechanics with advanced engineering systems. So as AI moves from the digital world into the physical world, robotics, wearable devices, biomedical devices, human-machine interfaces, what fundamental mechanics and materials challenges become most important? A very general question.
I would prefer not to say much about mechanics because I don't think the audience is interested in mechanics. But I'll do comment on, excuse me, I'll do comment on AI. During my 35 years of academic career, there are a lot of bubbles. Nanotechnology was a big bubble. United States government invested a lot of money. Nothing comes out of it. Nothing. Okay. There were a lot of material genome, a lot of things. They come every 5 years. Something comes, go. Something comes, go. But AI is real because AI has already penetrated society. Everybody uses AI. From that point, AI will have long-lasting value. That will change our society. But AI is also, lots of times, it's like in the virtual reality. So to bridge the gap between real and virtual, then we really need those sensors that can monitor monitor human and everything else 24/7. So those kind of sensor is what we engineer can contribute. Thank you.
Thank you very much, Professor Huang. So the next part, we try to connect with the 3 keynote speech. So in Professor Zhuang's talk, we have known how imaging technologies can illuminate biology at the nanoscale and also generate enormous spatial and also molecular data. So I think in general, what mathematical, computational, algorithmic, and physical foundations are needed to turn such data into real biological understanding? Well, I guess, because, the panelists on the stage are not from the biological world. So that be more specific. So for example, Professor Huang, so from an engineering perspective, how do advances in mechanics, materials, you don't want to talk about the mechanics, and device design shape the future of biological imaging and sensing? So from an engineering perspective, how would you see
From engineering point, one major advance is to make all electronics flexible that can be integrated with your human body. For example, right now you have to go to hospital to check everything, your EKG, your EEC, EMG, muscle activity, everything here. But with those flexible sensors, everything can be done at home. Think as an example, a patient in the hospital, and each night in the hospital, even without doctors, the hospital stay, the bill is $6,000. $6,000 just one night stay in the hospital. But if you can have those flexible sensors to monitor the patient at home, then there's a big change. And that's what engineering can do to the society.
All right, thank you, Professor Huang. So now, Professor Xu, you have mentioned, you want to actually apply your algorithms into, all kinds of applications. How would you see, those computational multi-time scale computation methods can handle high-dimensional biological data without losing the scientific meaning?
Indeed, and one of the, from computational mathematical point of view, we do hope that we have multi-scale algorithm to view this data from different scales. But the challenge is that we want to understand the mathematical meanings across the different scales. So we need reliable reconstructions and also the multi-scale algorithms that reduce the dimension. And but you reduce the dimension, but you don't. You reduce dimension, but you do not lose the size. So we do have various techniques over the years, but it's really. I often have seen Professor Zhuang's talk. This is all provide realistic examples and challenges for mathematicians to develop algorithms along these applications.
All right, thanks, Professor Xu. So in Dr. He's talk, he discussed the path from digital humans to humanoids. And once AI becomes embodied, it must deal with materials, mechanics, sensing, energy, uncertainty, and in particular, real-time control. So, Professor Targan, so what algorithmic principles do you think are needed when decisions must be made in real time, in particular under uncertainty and with safety constraints? Very hard.
I think this is very important, this whole area of security and safety. People are working on it, but they should spend much more time thinking about the issues. I mean, this idea of robots in every home is fascinating, but at the same time, these systems better be under control, because otherwise we can imagine all kinds of science fiction nightmares. And we know, of course, not only in the real world, but in the digital world, these systems humans have created many secure systems, but they aren't in fact secure. There are a lot of bugs, and these systems now have the power to find the bugs. We have to find ways to use these systems to find the bugs and fix them before bad actors get their hands on these powerful systems, and they could wreak havoc.
Thank you very much. So, Professor Luo, so again to you, how can optimization help embodied AI operate safely and reliable? And in particular, since you have been, promoting SLAE, so how SLAE is trying to contribute to this field?
I think safety security is a paramount issue. So from an algorithm's point of view, there's space for innovation, certainly. There's also data issue, data leakage. There's a hardware issue, hardware security, safety. And there's software stack security and finding bugs. Nowadays, coding agents are very powerful, and then they can also find bugs in the existing software system much better than humans can. So we should use these tools to secure itself. We still are the master of AI. AI systems should always be subordinate to human masters. So if we use it well, we can actually help achieve our goals and make our systems more secure. A simple thing. I mean, it's to just give people different privileges to access information. In existing computer systems, we can do that. So read, write, and all these privilege. But in the LLM, everybody can ask questions. So if I want to implement a large language model, AI system in my university, there's sensitive information that should not be reviewed to students, to faculty members, different levels of management. But we are all dealing with the same AI system. How do we secure that? Get it? Distinguish who's asking question and who should be getting what answer. That simple task, it's actually not easy to do. You think about it. So in SLAE, we have people working on all levels. We have 5 research centers, one of which is focusing on embodied AI, and the other one is in social science. So we focus on data and all these privacy issues and the ethics issues and security issues. So embodied AI, I agree completely with Bob. If we are not careful, these things can overpower physically human beings and then do crazy things. And then I don't know, if somebody else remotely control your robots, take over your robots, and can do a lot of destructive things beyond your... So we need to be very, very careful.
All right. Okay, thank you very much, Professor Luo. So Professor Fagan's talk actually reminded us that in biology, prediction is not enough. We need mechanistic understanding and experimental validations. So Professor Yao, in mathematics, truth is basically established by proof. And in experimental sciences, truth is often established by reproducible observations. How should we think about scientific truth when AI is involved? Is it possible that we can bridge these two mind sides?
If AI cannot be proved to be correct in many steps, the truth can never be established. It's as simple as that. I think the mathematics has been developed over a few hundred years. The modern mathematics is a whole subject is more than just fragment improvement. There's a global structure in mathematics. We look at geometry, we look at numbers. All this has a global structure, which I think engineers presumably do not realize. When we look at huge amount of networking, this actually already traditionally, that's what geometry would handle based on topological framework. Global topology tell you the global geometry, and then local geometry based on concepts like differential equations or curvature will take care of the local phenomena. And then put them together is a very non-trivial process, largely based on topological means. I think engineer, especially AI, should put all this together. Cannot just solve some local fragment problem and feel that you have finished what you do. When we try to find mistakes, we have also to look at global structures, not fragment structure. And this is actually very exciting to do.
Okay. Thank you very much, Professor Yao. So for the last round, actually, I want to ask all the panelists 2 questions. You can pick one of them to answer. So, well, it's actually the advices to the young generation. So the first question is, what is the biggest opportunity that AI brings to basic science over the next decade? Well, of course, it's a prediction, okay? But I believe many young generation actually are interested in that. The second question is that if you were advising a young scientist entering the age of AI, what is one fundamental skill or the habit of mind they must develop? Okay, maybe for this time, we can start from the middle. Professor Target.
I think it's important for students to develop curiosity and drive and direction and an ability to work really hard. I don't think AI changes anything. It's an incredibly powerful tool. One should learn how to use it, but not as a crutch, as a tool. And I think it's important to, why do we work on problems in the first place, us basic scientists? Because we are curious. The world is a beautiful place. Science and nature are wonderful, amazing, beautiful things. We're driven to study these things. This is what you need to do great science. The tools will change, but not this kind of human drive to do this kind of work. So find your own path, do what you love. This has always been the case. Large language models or any other kind of AI system is not going to change this anytime soon, I think.
Thank you very much. Professor Xu?
Maybe I want to share some of my thoughts based on experience from the use of AI technology by myself and my research group. First of all, I think the most productive way of using AI is to bring your own expertise into the subject. The more deeply you understand the subject, the more efficiently you can use the AI. And you ask the right questions, and also you should be able to tell if the answer is correct or wrong. Another thing I find is I'm telling my student, you treat AI like you want to be treated by your advisors. It's not like we should be patient. We should treat the AI like a human collaborator. We should be patient with the AI if it makes a mistake. We should not have the attitude, I got you. But rather be patient to ask the right questions and try it again. And from our own experience, that if you keep going and be patient, and you will see unexpected and productive results.
All right, thank you, Professor Xu. Professor Luo?
I think it's getting really late, so I better be brief. I think
all of us, including students, should learn how to ask good questions and be able to evaluate the solutions provided by AI critically and interact with AI.
All right. Thank you, Professor Huang.
I think for young scientists, number one, it's important to learn AI because it's coming. It doesn't matter what you think is coming. And I also know that in the company in the United States, the top performers are now those who can master AI. They really do job much better than the others. However, for scientists who want to do his or her own research, it's really important to have the concept that does not even exist. You build a new concept that cannot be searched on the internet. That's how you can outrun AI.
All right, Professor Yao.
Well, the development of science is not just answer questions. We need to create a way to understand nature, to understand what engineers want in the future. This means you have to ask right questions, creating new concepts that will help you to understand nature. I think we need to train AI working with humans together to look at what's observed in nature through experiment or through direct observation, or try to think philosophically what is needed. All this would be needed. Otherwise, I don't think human intelligence could ever be recaptured and understood. So,
Thank you very much. Well, thank you all for today's panel discussion. As Professor Luo just mentioned, it's getting late, so that actually concludes today's panel. So please join me in thank you all the panelists. Thank you very much. Please be seated. Thank you. Please be seated. All right. So today's conversation reminds us, that AI is not only a tool, it's also changing how we do science. But basic science still needs deep equations, deep questions, strong theory, careful validation, and real-world engineering. So in the age of AI, science is not about replacing human curiosity. It's about giving human curiosity new tools and also new power. So distinguished guests, this part of academic program has now come to a close. We will now move to dinner reception. So there is an old Chinese saying, 有朋自远方来，不亦乐乎. It's a joy to have friends to come afar. So tonight, we brought together by science and we are connected through conversation. The dinner will be served in lecture halls B and C. Please proceed to the dining area and continue your discussion in a relaxed setting. So at 8:15, we will continue with a wonderful special performance. Please return to this hall in a time and enjoy the performance with us. So that's all for